So just get right into it. So just a little disclaimer first that most of what I'm going to talk about is not my work specifically. I may sometimes involve in some of it, but I'll try to do it justice. So first of all, what is this theme? Basically, we're thinking about cancer and its relationship to systemic perturbations throughout the body. So, and we're going to use that. It seems very vague, but I'll get more specific in a minute. But we want to see if we can use that for strategies to do things like early detection, prevention, and treatment. Okay, so roughly what's ended up happening in this theme is that there's kind of been two main approaches that have been applied to investigate systemic responses. So first of all, causal analysis. So this is just trying to look at mechanism. So, and specifically, there was a focus on inflammatory markers. So inflammation is known to be related to cancer risk. And then the question is that just sort of a byproduct of something else that's causing cancer, or is it's possible that the inflammatory markers themselves are somehow related to cancer risk? And then the method tends to be Mendelian randomization to investigate that. And then the second part, which I'm more involved in, is biomarker discovery for predicting risk. So here we're using typically molecular data derived from blood samples. So that's the systemic part. So we're getting factors that are circulating through the body, and then trying to see if we can predict cancer risk from it. So we're not worried about causation or mechanism. We're just looking at what are we predicting, and then hopefully we can find some independent data which we can validate our models. So it's more of a machine learning approach. Okay, so first highlights. I'm gonna select 3 highlights from the related to causal results, and then roughly something similar for the prediction. So I'll start with the mechanism or causal results. So the first highlight is that we now know that some cytokines do seem to influence cancer risk. So what was known before was that inflammation is associated with cancer risk, which I just mentioned, and that cytokines are inflammatory signaling proteins. So does the abundance of cytokines in blood influence cancer risk? So our partners from Greece led this project, and they applied Mendelian randomization to 47 cytokines and 5 cancers, and they found a causal role for 6 of the cytokines. And so, without going into great detail, but they found evidence for a minority of them, but as we know, Mendelian randomization is not a method that's going to tell you everything. Like, it can tell you what possibly has a causal effect, but it's not going to necessarily tell you what doesn't have a causal effect. So at least we know that there's some evidence that 6 cytokines seem to be involved in causing cancer. And so the impact of that is it identifies possible targets, possibly for cancer prevention or possibly for treatment. Secondly, so following up on this, James Yarmolinsky led this study asking, well, let's ask more broadly, so not just cytokines, but other inflammatory markers. Do they appear to influence cancer risk? And the conclusion there is that there didn't seem to be a huge amount of evidence. So they looked at 66 inflammatory markers and 30 cancers beyond just cytokines. And found out that of those 66, 4 markers were linked to breast, pancreatic, and skin cancers. And furthermore, there was little evidence for any link for 22 of the 30 cancers. So this gives us a little bit more information about what we can expect from inflammatory markers. So there's potentially saying that maybe this isn't, some of these might be useful for looking at in terms of prevention or for treatment, but this isn't going to be a gold mine where you're going to find a huge amount of things that you can use either for prevention. And then finally, this is led by Emma Hazelwood. So this was published very, very recently. Well, actually not published yet. It's on preprint as far as I know. So here, we're asking, we're getting a little bit more subtle. And instead of just asking about adiposity or BMI, which is sort of a, body-wide fat, here we're looking at more specific fat distribution. So if you have a adiposity higher in certain parts of the body, does that give different information about cancer risk? And so here they once again applied Mendelian randomization to 5 adiposity distribution traits. So abdominal, subcutaneous, visceral fat, femoral fat, liver, and pancreas fat deposits. And then how do they relate to 12 obesity-related cancers? And as you can see, the table there, for certain deposits, you actually get an increase. So the increased fat deposit increases the risk of the cancer, but not always. Some of the fat deposits actually give a decrease in cancer risk. So this is suggesting that maybe some of these things that we've been studying previously need to be revisited in terms of how adiposity relates to cancer risk. Okay, and so just as following on from that, we're actually, Richard mentioned this earlier, that we have this successful application to look at obesity-related cancer. And so it's going to basically follow up on that study that I just mentioned, among other things. But of course, we'll look more into how these different deposits relate to cancer risk. And then, of course, there's other work packages related to what I'm going to talk about next, which is about making predictions about who's going to most benefit from certain interventions or treatments. And then, oops, there's a little mistake there. It's for package 3. It should be at the bottom there. Actually working on specific ideas for interventions, and so what's going to be most effective. So there's 3 parts to that, and I'm sure you'll hear more about it during the day. All right, so moving on. So here we're moving on to prediction. So leaving behind mechanism here, we want to do is if we can see if we can predict cancer earlier, or in order, because we know that cancer, when it's predicted or detected earlier, treatment is usually more successful. So here the question is, can proteins in blood plasma outperform questionnaires for estimating lung cancer risk? So the study used 6 cohorts, 470 cases for training, 154 for testing. And then, of course, they're comparing against the questionnaire, which is the PLCO test, which just asked about, smoking history, cancer history in the family, things like that. And then compare that to a model which is derived from 302 proteins measured in all of these blood samples. And the blood was collected 1.7 years before diagnosis. So if the model is successful, then it's potentially helping us identify people who are going to get a diagnosis or at high risk of getting a diagnosis. It's going to give earlier information. So it was trained using an elastic net, in case you're familiar with that. And what came out was a 4-protein model, which we'll talk more about later in the day. The performance, so at specificity 86%. The protein model outperformed the questionnaire. So at the protein model had 49% sensitivity, whereas the questionnaire had 30% sensitivity. And if you're familiar with area under the curve plots, you can see there that the 4-protein model had area under the curve 0.75 compared to 0.64 for the questionnaire. And so the impact of that is just that we now have a simple blood test that can identify high-risk individuals earlier and more accurately than the questionnaires. All right, and then we'll talk more about this in the afternoon, but we're actually putting together a funding proposal to follow up on these things, and we'll be adding DNA methylation to that study or to that proposal. So one of the things we know is that DNA methylation in blood actually, we already know that it outperforms questionnaires, or we have some evidence for that. DNA methylation is also interesting in that it's more stable than protein abundant. So whereas protein abundant, so proteins will go up and down depending on what someone's eating or whether they're sleeping or whatever. Methylation does some of the same things, but tends to be a bit slower to respond. And so we're thinking that it might provide potentially longer term predictions of lung cancer risk. And so if you put the proteins and the DNA methylation together, you get something that kind of works along quite a short scale and also a longer scale. But that's to be shown. The next one here. So what we wanted to do was we run into this problem often that with molecular data sets, the numbers are small. And so we want to use things like, methods from artificial intelligence, machine learning. But for those models, you need high amounts of data, large amounts of data, lots of samples. And so if you were dealing with like a rare cancer, you're going to have small sample size. Also, molecular measurements tend to be a little bit expensive. And so then that constrains the sizes of the data sets. And so what we wanted to ask here was, can we use some information from large cohort studies that have large data sets, large sample sizes? Not necessarily the disease we're looking at, but we're going to use that data to tell us about the molecules. How do they relate to each other? So we get lots of information so that if one protein, for example, goes up, we know that another protein tends to go down, which we don't get as good information from a small data set about that. And so the idea is to train machine learning models, artificial intelligence models, or here we call them deep neural networks as an example. Train those in something like UK Biobank, where there might be 50,000 people with protein profiles. And then we can go to a smaller data set where we have the cancer of interest or the cancer cases of interest that we want. So in this case, it's head and neck cancer. And we have, so compared to 50,000 in our initial training for our deep neural network, now we go to a small data set with 156 head and neck cancer cases. And so what we found, though, is that the model which benefits from UK Biobank and from the large data set, it actually outperforms models that are sort of more trained in a more traditional way within just the head and neck cancer, the small data set. So area of the curve is 0.88 compared to less than 0.77 for traditional approaches and models. And so then the impact of that is that we hope that we can sort of generalize this approach and we can start building large AI type models from some of these, UK Biobank and other really large international cohort studies. And then once those are created, we can then go to individual diseases, including rare diseases, and produce models that perform well. Okay. And so, as I said, we're now trying to generalize this. So we're actually building a DNA methylation model at the moment similarly from about 50,000 cases, and we'll be initially applying it to a prostate cancer case control data set with 125 cases. All right, and that's it. So thank you for that. Brilliant.
I was going to ask, that the DNA methylation, and presumably you can then do that for metabolomics, proteomics, anything. Yeah, that would be really good, because then we can apply it to things like the PROTECT study, where we've got a few low numbers of deaths, and so on. Yeah, that's absolutely fantastic. That methodological work. Any questions from the yeah? Yes, Evie.
Oh, I know we're primarily dealing with adult cancers here, but can these techniques be also applied to children's cancers?
Yeah, no, I don't see any reason why not. I guess what we lack at the moment is that most is these large data sets. So there's a lot of, large adult cohort data sets. And so that's always been a problem, is that we have less data for children. But that can be changed. We just need the money to do that.
Thanks, Matt. So have you, so this is a little bit outside of what you presented, but have you been giving any thought to sort of the dementalization approach and prediction? So to use genetic markers of risk-informative biomarkers that are not false, and sort of remove the genetic noise that occurs because they're genetically influenced to improve the prediction. Have you been given that any thought? And also perhaps in the context of ICEP?
Yeah, so we've definitely given that a thought. George has definitely given me that thought.
It's not preprint has never been published because it doesn't work. But we've now worked out why it doesn't work, which is really quite interesting. I'll talk to you about it. I'm going to talk to you about it. The only published thing suggesting it does work is the Nicaturis piece on PSA. And it works very, very slightly, but in general it doesn't work. Which is why preprint remains a preprint.
Thank you very much. We had a question. David, you have a question, and then Athena.
Good talk, thank you. Going back to the stock of information at the start. So where you've got generally an association between information and various cancer types, but then the DNA is 60 on protein panel and found very few cytokine or not associated. So what are we missing? What's the sort of hypothesis for why we don't see more of the classified inflammatory markers associated with cancer risk?
If any of the authors on that paper here, but I suppose, one issue is that we're just looking at, when we take a blood sample at the moment, when we measure proteins, we're just measuring, we're getting proteins from all over the place, right? So it's possible that we're just getting a mix of signals, and as a result, it might be that there's one specific cell type that would give us the protein abundance that we need to know, but it's sort of getting obscured. That's one possible.
Yeah, so any signals just sort of diluted out against the bulk systemic thing that we're looking at rather than what's contained locally.
Yeah, that's the one thing that comes to mind. And we're trying to address that in various ways, but it's expensive and difficult because you have to then drill down into exactly which tissue, which is difficult.
Matthias, do you want to say something?
Yeah, I just comment on that. I think the sort of idea of looking at all these cytokines and trying to tease out exactly which ones are sort of causing disease is a huge challenge in its own right, because there's like hundreds of them, and at some point we run out of data to do anything that is sufficiently robust. So I don't know if it probably is solved or how to understand that particular question. Even if we're convinced that information is important, even in the important sense, understanding it precisely, I think it's just very, very challenging.
Thank you. We'll have one last question from Athena.
Back to your previous one, where you've got your area of the curve, and we use VLCO, which is a prostate lung cancer screening trial from months ago in the US. And I just wonder if anyone who's chosen that one, I'm seeing how representative you kind of then went on to imply that all questionnaires are rubbish and all method was now better. I suggest there might be further better ways, but the questionnaires more than cohorts, and inevitably the nature of a trial, you've recruited a subset of participants to then be giving the answer to those questions.
Should I comment?
Sure.
Yeah. So, there are a lot of sort of risk models for lung cancer, and we have studied these extensively, and the PLCO is the one that is basically the most robust, I think. So that's just one of those points. whether the methylation markers can we improve the count when we get to proteinuric. There's some slight hints on that. But it's quite clear that we can at least replace it. And there might be some implementation settings where that is quite important. And we're going to show some data from now completed studies that shows also that the protein biomarkers can add substantially to that. This concept of using just a blood test to test whether someone should get screened is quite compelling in specific settings. So